All right. So obviously this is the second half to the biology majors class, the 1407 class, and you know, we're using the same textbook and the same lab manual. So if you have those, you actually see as we go through the schedule which exercises we're going to do in the back half of this book. And you can see which chapters we're going to dive into in the back half of the textbook. But before we get into any of that type of stuff, I wanted to kind of go into, in this first meeting, a little bit about how we're going to approach the second half of this class and how it's different from 1406. I think one of the first things I put on the board in 1406 was this scale of nature pyramid where we spent a lot of, like, just like last semester, this semester is broken into 4 units. And we tackled this pyramid last semester where we spent a unit talking about chemical bonds and molecular bonds, and then we spent a unit talking about organelles and cells. We spent a lot of time inside the cell last semester. And I think right by the end of the last semester, again, everybody that is here, I remember we talked about evolution and speciation at the end of last semester. So I don't know if you had that at the end of year 1406. Sometimes it's thrown in there, it's not. Because those concepts of evolution and speciation are going to come back, but we're going to be looking at them from a little bit larger scale. So we made our way all the way up to organisms last time, and we were talking about examples of different types of organisms. We called those species. We defined species using our biological definition, and we talked a little bit about the forces that create new species, that push species in different directions and cause them to adapt to different conditions. Basically where I'm going with that, and where you can see where we're going with this, we tried to draw a connection. We had a lab last semester where we put different habitats down, right? And we spread those beans on them, and we were predators, and we were picking out those different beans. And we saw that there was this connection between habitat diversity and species diversity. And so really what we're going to do in this first unit before we get into examples of different species, I want to kind of set some background information up about ecosystems and biomes, and really how the ecosystem and the habitat itself really predicates or determines what type of plants can live there, and that determines what types of animals can live there. So we're going to kind of take a broad approach here. And again, we start with organisms on this pyramid. We're going to work our way up to, this is kind of one of these nested doll situations again, where multiple organisms in a group, we refer to that as a population. Our class is a good example of a population. We're all the same species in the same area at the same time. A community, just to define this term real quick. Again, I feel like I'm getting into Monday's lecture already, but I'll peel myself away from this. Just to define community, this is going to be multiple populations working together. So we are a population, and I've got the blinds closed, but sometimes you can see that wasp nest that's up there. There's a bunch of wasps in there. That's a population of wasps. And I'm sure there's mice running around in the ceilings, or maybe some bats or something like that up there. So if we think about all the different populations of mammals that are around us here, that refers to our community. So a population, multiple populations, all interacting together. So that's fine. The level above that, we're going to see ecosystems. I'd kind of mentioned this earlier. Let me just point out, when we get to the level of ecosystem, we are considering 2 things. We're going to consider biotic factors. And again, we'll talk more about this on Monday. And the ecosystem also includes abiotic factors. Abiotic factors would include things like the temperature of the room, how much sunlight hits that side of the building. All of these non-living factors or components. We'll see different ecosystems like Arctic tundra. We saw desert environments last time, or even wetlands like the Louisiana coastline are all examples of different ecosystems. Above the level of ecosystem, we're going to see what's called biomes. And depending on what textbook you use, there are anywhere between 8 to 16 different biomes, depending on how nitpicky you want to be. Biomes, examples of different biomes, like I just mentioned, I'll use a different color in there. There could be things like wetlands, or maybe Arctic tundra, or maybe desert environment, savanna, grasslands, right? And whether it's Arctic tundra, grasslands, or wetlands. That has a lot to do with rainfall, temperature. In fact, one of the things we're going to see on Monday's lecture, we're going to get into a little bit of a climate discussion or weather, just to talk about what causes that. How is it that these different types of biomes, came to be in the first place, and what kind of pressures that puts on the animals that live there, right? It's all about selective pressures. If we go back to last semester. All these different biomes are found just in the only place that we've found life so far in the universe, and that's here on Earth, which we refer to in the textbook as the biosphere. So this is the pyramid that we're going to tackle this semester. Obviously, we will start once again towards the bottom of that. But I just wanted to compare a little bit of the scale of nature about how this is different. I've got a couple of slides that we're going to look at today, just to once again kind of explore that difference in scale of nature between 1406 or 1407. But as always, interrupt me if there's questions about anything, or if something I say triggers a thought in your head. Okay, so if you have the syllabus in front of you, let's just make sure that we're all in the right place. This is the 1407 class. This is the Monday, Wednesday, 12:30 to 1:45 lecture, and our labs are going to follow the lecture, is what I will say there. This syllabus says that it shows that we have lab every day, but that is not accurate. Instead, we have this block of time, is what I can say. From 12:30 to 3:15, we have that block of time. Some days, we will spend most of that time on lecture if it's a day that we don't have lab. Some days it might be a point in the semester where we have we've gotten ahead on lecture, then those days might be mostly spent on lab. So as you know from last semester, we typically have one lab a week. I'll show you the schedule. Our first lab isn't for like 2 weeks, so there's not a rush on the first one of those. There is just one combined score for this class, so there's not like a separate lecture and separate lab grade. All the points just go into one big pile at the end. What else can we say here? I want you to be able to contact me if you have questions. There is my email. That's probably the preferred way to contact me, but for some reason, if I'm in between buildings, on Tuesday, Thursday, I start off in Taylor. That's where my physical office is located. So I'm always there from 1 to 2 on Tuesday, Thursday, just hanging out. I know that might not help anybody here if you're primarily around Hutto. So I'm also available to virtually meet in the Teams space at whatever time works for you. I have evening classes Tuesday, Thursday, but I'm up for meeting early or late, whenever works for you on that. Even though I don't have a physical office space here, you can typically find me in this room or across the hall in the A&P room. There is also a couple of phone numbers that you can reach me if there's any kind of emergency. The next thing is the course description. I've kind of been dancing around this already. This being the second half of the class is one that we finally get out of the cell and we start to look at things at more of a population level. In fact, we're going to see a lot of diversity a broad range of diversity, I should say. This is one of the great things about this course. I like it because, again, we get to zoom out, we can get our hands on things finally. We can see things with the naked eye. But the trick, I guess, or the trouble with this class is that we've only got 16 weeks, right? And that's not going to be enough time to accurately cover all of the things that we're going to introduce, basically. D2L's not working.
I went to Tuesday classes.
Oh, no, you're good. I know. I keep thinking that it's Tuesday myself, but no problem at all. You're right in time here as we're just going to go through the syllabus. I'm trying to point out that the second half of this class, we finally get to see things that we can get our hands on. We're going to start with small things like prokaryotes and protists. We'll have to use microscopes to see those. Right now, prokaryotes and protists sound maybe similar to you, but hopefully by the end of the semester, we'll be able to distinguish the difference between protists and prokaryotes. Both of those are tiny, so if you are by the end of the semester, you should figure out whether you are interested in microbiology, whether you are interested in mycology, which is the study of fungus, whether you're interested in botany, studying plants, or maybe it's some type of zoology, invertebrate zoology or vertebrate zoology. We're going to skim the surface of all of those. You could spend an entire semester on each one of those, and we don't have that. So we're going to sample each one and hopefully help you decide whether you want to go towards a certain field or away from a particular field. The first unit, however, unit number 1 in this class, we're going to do a little bit of background information just about classification and how to draw evolutionary relationships. So there's a lot of, once again, systematics and evolution that come in. There are the same textbooks and lab manuals. I'm guessing you still have those from last semester. What else is here? The next thing that is important is the grading scale. It's just about identical to last semester. You can see there are 4 lecture exams where most of the points come from. Then there are, once again, the lecture quizzes. There are 8 lecture quizzes. I'll pull up the class schedule, which you can currently see on the back of the I think, second to last page on your handout. It shows that there are 4 units. Each unit has 2 quizzes associated with it. In those quizzes, you can see carry 35 points with them. Just right off the bat, and everybody already knows this, but I'm not a big fan of extra credit, but I'm a big proponent of regular credit, right? And here is the regular credit, and some of these points are easy to get, like the quiz points, as I won't be able to show you because D2L is not working right now. Maybe it is. If it does work, I'm just going to edit that out on the clip, and it'll just magically work whenever you watch this later online. But you will see on D2L, it tells you the dates of availability for each one of these quizzes. Remember, the quizzes will be up on D2L for typically 7 days. And within that span of 7 days, you can take those quizzes as many times as you can. And if it were me, I would, since you can take it more than once, I would take it immediately once, find out what the questions are, look those questions up, and take it a second time and get all those points. You'd have to do it at least twice that time, but you could make sure that you're making sure that you get all of those quiz points whenever you run into an exam and might not get all the points that you want there. It's good to make sure that you get the easy points. Most of the points come from lecture, obviously 700 available points for lecture. At some point in the semester, I'm going to bring another class activity that's going to serve as our core assessment assignment. I don't know which unit I'm going to bring that out in yet, but whatever day that is, it'll be a class activity. It'll be worth 20 points. More info on that as we get closer. But the second box down here is lab. You can see there are 300 possible points available in lab. Most of those points come from the lab activities, just like we did last time. They come from this lab manual. Again, there is participation involved in that, so as long as you are here and going through those activities, that will factor into some of those 20 points. But in addition, the new thing here is that there is no lab practicals in the second half of this class. Remember last semester we had these lab practicals that were worth 50 points apiece. And I'm always critiquing things, and what I didn't like about that is that we would go about 6 weeks and then we would have a practical over something that we talked about 5 weeks ago. And it was a stressful semester, so sometimes it's hard to remember what we talked about 5 weeks ago. So to just kind of ride that wave of information without it getting too overwhelming, every time we have a lab in here, there is going to be a separate quiz that's going to show up on D2L. It's going to be under the quizzes tab, just where the other quizzes are. It's short. It's only going to be probably 10 questions at most, and it's going to be focused on what we covered in lab that day. Unlike last semester where I was stuck inside the cell talking about a bunch of different processes, when we get into talking about plants and things that we can see, I want to take campus tours where we're going to go walk around and we're going to collect campus plants and we're going to learn the names of all those campus plants. Because it turns out these are very similar, if not the same plants that you have in your neighborhoods. And if we can learn what's here, you'll see these all over the place. And those are the types of things that are going to show up on post-lab quizzes. We should be able to do some dissections in here when we get into animals, vertebrates, and invertebrates. We'll dissect, obviously, flowers, different plant parts. Those are the things that I'm going to take pictures of and put on the post-lab quizzes. So as long as you were there or have done whatever it will be the relevant makeup lab portion of that, you should be able to answer those questions. Just like regular quizzes, they're going to be available for typically 7 days or so that you'll have a chance to take them. I think that's the main difference that I wanted to mention. Everything else on here is going to be something that you could pretty much get on your own by reading. We've got formats of the exam, which tend to be mostly multiple choice, but I might put some short answer questions or matching or true false on there. Obviously, we can't have cell phones during exams or leave the classroom or any of those types of things. Unfortunately, not that anybody in here has ever done anything wrong, but previous experiences have made it so that when we take an exam, if you've got the exam date, so just know when you're ready to walk into the exam. If you need to go to the bathroom beforehand, go to the bathroom. Again, that's pretty self-explanatory on there. Online quizzes, those are going to be found under the Quizzes tab on D2L. I will attempt to see if that's working again in just a second. Maybe I'll be able to show that to you. If not, it sounds like everybody in here is familiar enough with D2L to be able to find the Quizzes tab. The only other thing that is down here is that note that I had mentioned about COVID absences, and you could really even expand that to just bad weather or whatever it might be. Remember, since there are already the recorded, I'll say recorded lectures in quotations because it's just me talking through the slides. Those are already up there. If you miss a class, that might be sufficient to catch up on what we talked about in here, but to really make that better, I'm going to supplement that with the actual recorded in-class time. So are there any questions about the syllabus so far? You have 2 additional pages past that, and one, there's some pictures. One is the thing that looks like this. It's our lab schedule. And you can see this week there's no lab. It's basically a wellness check this first week. I wanted to make sure that we're familiar with how to navigate the course, how it's going to go, that we all survived the winter break. Having survived any kind of situation like a fall semester, it bonds everybody together. So I'm glad that we're all back. I'm glad to see all the familiar faces. And I want to make sure that we have another fun time this semester. So our first lab, you can see that's going to be exercise 12 in the lab manual. It's not going to be until our week 3. Then there's another week where we just don't have a lab. And the reason I put a no lab right there is because the first unit of lecture, we're really just talking about, like I said, these broad biological concepts and how we name and classify things, how we draw evolutionary relationships between things. And then once we get into Unit 2, we get our hands on those things, like the prokaryotic cells, the protist and the fungus, and then after that comes plants and animals. The second half of the semester, the labs become very intensive. But it's a little bit slow to start just because in unit number 1, we're not really talking about things we can get our hands on yet. What else? And again, there's no practical, so there are just going to be post-lab quizzes that follow each one of these. So there's the lab schedule. I'll get that out of the way. I'll point out that you have in front of you a lecture schedule that looks like this. This is the most updated version of this. And what I've done on this one is under the online assignments, if you were part of the class last semester, there was a little bit of confusion because I believe on the syllabus, I was calling these things homeworks. But when you went to D2L and clicked on the quizzes tab, they were called quizzes, and it was a little confusing. So I've tried to clean that up this semester. Since there are going to be quizzes based on lecture information and then some quizzes based off of what you've done in lab, I've made sure to name those quizzes either lecture quiz or lab quiz. And when you go to D2L, you will also see the same naming arrangement. Currently, if you go to D2L, you shouldn't see any quizzes because your first quiz doesn't pop up until next week when we get into actual course information. That will be a lecture quiz that pops up that lets you know it's going to be over lecture information. You can also see the exam dates and everything in there in terms of important dates. I just wanted to show this same thing, but in a different version. There's a slightly more dated version. It's probably literally 3 hours older than the one that I just showed you. You can see I had not yet added the post labs, or even named the quizzes different between lecture and post lab quizzes. So it's a little bit older version. But what I did while this was just sitting in front of me, and I don't even know if you can see that. I'm going to kill that light too, just to see if this is even going to be visible on that computer. But what I added to this is really just, let's see if I can zoom in a little more. This is just my way of kind of breaking down this semester. I had talked about it's a class that covers a broad range of topics, and you could spend entire semesters on things that we will just spend 1 or 2 class periods on. So it's hard for me to decide what to leave out and what to leave in when we tackle these things. But here's basically how it goes, just to kind of give you a roadmap of what we are about to go through. Here we are today. We're making sure that we introduce the syllabus. If there's any questions about last semester's information, evolution and speciation, obviously feel free to bring those things. I last semester had shared a lot of my misconceptions that I had whenever I was going through school and trying to learn about this stuff. I come from a real small town where I was taught, again, by coaches that were more concerned about winning a game than teaching me any kind of science. So it was hard for me to find places to discuss these types of ideas. And obviously I'm glad that we have one of those here. But so that this makes sense, I wanted to spend the first unit of this semester kind of addressing, once again, some more of these misconceptions, which I will do. In fact, before I jump directly into examples of that, let's just look at how I've broken down the schedule. So Unit 1, I'm basically just saying broad biological topics and background information that we need to understand in order to make the second part of this biology class make sense. For example, you can see we're going to be talking a lot about deep scale time. So we're going to, right off the bat, one of our first new lectures, and I apologize if the screen is blocking you here. A week from today, we're going to do our Earth history PowerPoint. You're going to get a handout about geologic time scale, and we're going to have to burn into our brains the eras, the dates, and the time frames of certain important periods in that geologic time scale. We're going to be referencing those the entire semester when we talk about when plants or when fungus made their way onto land for the first time, or when amphibians or reptiles or mammals first started to show up. So right off the bat, we're going to get us a good foundation about geologic time scale, Earth history, and then you can see the second half of unit number 1 is all about showing these evolutionary relationships, how we classify the organisms, and then how we show those evolutionary relationships is the systematics part. So once we kind of have that foundation down, then we start looking at examples of organisms, and this is really when the labs start to become more intense. The first half of unit number 2, if you are interested in microbiology, you will be happy because we spend a couple of days talking about prokaryotes and viruses, and then we spend a couple of days talking about protists. Again, right now, it might not be obvious what the difference between those two are, but once we get through this unit, we'll have plenty of examples of different types of protists and different types of prokaryotes. Often single-celled organisms that we're going to have to use microscopes to see. So this is the world of microbiology. Then at the end of Unit 2, we squeeze in a couple of days. If I'm careful, I might be able to squeeze in a third day of information about fungus. Again, we could spend an entire semester on mycology, or the study of fungus. We have 2 days to talk about all the examples of different types of fungus. So we start to throw things together there in unit number 2. Unit number 3 is all about plants. So we're going to get our hands on a lot of stuff there. It's nice that we won't need microscopes. We can go get our hands on plants. They can't run away from us, so it's easy to go collect these things. We should be able to get out and start to take trips, at least move around the campus when we get into plants. Again, you can spend an entire semester on this, which would be a botany class. Then we finally get into Unit 4, which is where I'm kind of partial to because it's the unit of animals. And maybe before I even do this, let's try this. Let's name the first 10 animals. I don't know if we did this last semester or not. Name the first 10 animals that come to your mind. I'll cross them off as we name them. Just what do you think?
Dogs. Bear.
Oh, so I got a dog and a bear.
Yes.
I got a horse and a cat.
Tiger.
Tiger and a monkey, I think that's 6 so far. Parrot, that's a good one. Coyote. Pigeon. I heard a kiwi and a barracuda. So we are now over the limit of 10. But I'll point out that every one of those that we've mentioned, those are animals that have a backbone. Those are vertebrate animals just like us. And it's interesting that when we think of animals, we automatically think of animals like us, vertebrates. But it turns out that vertebrates make up less than 5% of all the animals that are out there. Like over 95% of all the animal species that have been found so far are animals that don't have a backbone. This is the world of invertebrates. That's why we have so many days set aside to talk about the different types of invertebrates. So invertebrate zoology is an extremely large field. But, you know, like we just said, when we start to think about animals, it's all about vertebrates. So there's where the amphibians and the reptiles and all the mammals are located. And I'm a mammal person, so we're all mammals, but that's where I'm kind of had a lot of interest or focus. So maybe I can squeeze a couple of days of vertebrate zoology in there. Again, we don't have enough time to cover all the details that I would like to cover in each one of these subjects, but we will have enough time to introduce us thoroughly to each one of those subjects. So by the end of this, you know, you'll have a good idea of where you might have more interest. But again, what I have done in the past is tried to keep all of the course information in one of these binders that has a see-through little cover slip on the front and the back, and I can put my lecture schedule on one side and the lab schedule on the back, so I can immediately just glance at this and go, okay, no lab this week, I'm okay. We're talking about, you know, just review and syllabus stuff today. You can see where we're going next time, which is I'm going to pull up some slides. We're going to look at examples of biomes and ecosystems and communities, just trying to once again give a good foundation and address any kind of misconceptions in Unit 1 before we get any further into the semester. How are we doing so far? Does everybody feel okay? Any questions about the syllabus or schedule? Then we are almost done. I just wanted to say something about, just to give you an idea about the systematics in evolution. And I feel like we had one of these discussions somewhere. This is popping into my head from last semester. But just to give you an idea of the types of misconceptions that I had. And you can, or maybe not, share with me if you've had similar misconceptions about these things. But we're going to be drawing a lot of evolutionary trees. This is the systematics part of it. And it tries to show evolutionary relationships. So here's an example of things that we're going to be doing. I'll do this with mammals right now, but we'll be doing it with all different types of organisms. There, like at the end of last semester, we said there were, now it's working, okay. So in the video, I'll just edit this to jump to right here real quick, but let's just quickly show where to find some of this stuff on D2L. I won't spend a lot of time because I know everybody in here is relatively familiar with it. You can click on that little box of tiles there, and then you'll want to come down and find our class, which is one of these. It's this one. And the landing page doesn't ever really take you to anything. You'll have to click on either the Content tab or the Quizzes tab. These are going to be the main two tabs. Apparently, currently, there's something under Dropbox which shouldn't be there, so I'll remove that. Pay no mind to that. The Quizzes tab, if you were to click that right now, I don't think that you're able to see anything because these have not yet hit the date at which they show up to you. But the first quiz, which is all about Earth history, is going to show up on Wednesday, you can see. And on here, you'll be able to see the availability date, when they start and when they go away. Obviously, that's still under construction. The other tab is the Content tab, where you can find everything. Obviously, to the left is your little navigation bar. You can quickly go to class information, which is where you can find the syllabus, the class schedule, and the lab schedule. Those, you got copies of all of those today. That's what I handed out. And on the top of the syllabus is where you can see my office hours. So I'm just going to jump over here to unit number 1. You can see that on the top of unit 1, I went ahead and put in the same PowerPoints that we ended last semester with if you just wanted to have a review of evolution or speciation. Again, I'm about to talk a little bit about that. But the new stuff I'm going to say is really when we start with the geologic time scale in this History of Life PowerPoint. Just another thing to mention, since we finally got the D2L to work, I've noticed that other instructors will post PowerPoint slides in a PDF format, which what I've noticed is that if you try to print those off, it's hard to get them like multiple slides per page. Maybe there's a way to do that and I just don't know how to do it. But I post PowerPoints in the PowerPoint presentation format because, at least as far as I know how to work these things, it's easier to print PowerPoints where you can print them in multiple slides per page. Here's what I mean. Just as a tip on how to take notes or prepare for exams, you download one of these PowerPoints. However you want to get to your print screen, I'm just going to go Control P here. Instead of just going straight over to print, remember to look for that drop-down box that allows you to print multiple slides per page. And my tip is always to print 3 slides per page like this for a couple of reasons. One is it gives you room to write notes down when we're looking at particular slides. Instead of printing off the slide and having to write your notes on the page, this kind of gives you a little bit more of an organized way to write notes. And also, the second thing that it does is it shows you at a quick glance. If you were to do this for all of the lectures that we have, you know, a day later, if you're just flipping through your lecture notes, you can see, oh, I wrote a bunch of notes on that particular slide, and I didn't write many notes on this slide. So it tells you at a quick glance, we spent a lot of time on that slide. This is an important slide for the exam. This one might not be as important. You can see the last part of Unit 1 is all about systematics and showing these evolutionary relationships. And that's where I was about to mention something from last time. We said at the end of Unit number 4 last semester, we were pointing out these evidences of evolution. One of them was molecular data, molecular comparison between different species. Right? I've already erased this chart, but when we start to compare at the bottom level different types of organisms, how do we compare species to one another? One difference, one of the things that we can use is molecular data. And really, it's only been in the last 20 years that we can start sequencing the DNA of different species. And they found that our closest living relative, I remember reading this, 20 years ago or so, our closest living relative as humans is chimpanzees. So, thinking of it in a linear way, how I remember first thinking about this. Okay, we've got chimpanzees. They're the closest living relative to humans. Right? So we've got humans over here. And I remember thinking, okay, well, if evolution is working, then where was it at some point in the past that there was one chimpanzee somewhere that evolved into a human? Or how long ago was it in the past that we had maybe a jump from chimps to human? And this idea of thinking about it going from chimps to humans is incorrect. That's not the right way to think about it, right? But it just shows that I was taught by mostly coaches, but that's the incorrect way of thinking about how speciation or how evolution makes jumps. Instead, how we're going to show this relationship between chimpanzees and humans, right? Like the literature says we share 98% of our DNA with chimpanzees. There's only 2% difference genetically between humans and chimps. And that's right now let me add one thing to this chart, and that's going to just be this vertical series of lines that we're going to use to represent time. So up here at the top, this is going to be the present. Right, we'll just say 2022. Here we are in the present. And the other lines here, this is going to be like the dirty clothes hamper that you have where the clothes at the bottom of the dirty clothes hamper have been there the longest, right? So this is geologic time scale. The further down we go in the rocks, the further back in time we go. So we'll say this might be something like 3 million years ago. This one might be 7 million years ago. Down here we're at 10 million years ago, maybe 30 million years ago. Right, so we'll have to put some kind of measure of time in here when we start to talk about these evolutionary relationships. And it turns out that here in the present, right, in 2022, we've got humans, we've got chimpanzees, we've even got these other things called bonobos. Right, and you find all of these roaming around here in the present. But if we start to think about evolutionary relationships, it turns out that if we were to go back I'll just go to here. And I'll jump over there. So if we were to go all the way back to dig in the soil to find rocks that are 7 million years old, right? If we go, you can do geologic studies to find where rocks are of different ages. And if you find rocks that are about 7 million years old, and we go back and we find fossils in these rocks about 7 million years old, you won't find humans, you won't find chimpanzees, and you won't find bonobos. Instead, there is one population of upright apes. It's called Australopithecus. We don't have to remember the genus Australopithecus, but there is a group of a population of organisms, upright apes, that over time went their separate ways and adapted to different conditions. And over the next 7 million years, it led to differences that we now see as between chimps and humans. So it was wrong for me to think that at some point chimpanzees are now going to, some of the smart ones might evolve into humans. Instead, you can think of that chimpanzees and humans shared a recent common ancestor back in the past, and since then they have been diverging, and they will continue to diverge. In fact, even humans on our own branch, we are continuing to diverge. Just like the other branch between chimps and bonobos continued to diverge. There was another molecular event that occurred at 3 million years ago. This was between chimps and bonobos, where bonobos and chimpanzees are more closely related to each other than either are to humans. Right? So we'll call this examples of sister taxa. They are more closely related to one another, and the differences primarily between the bonobos and chimps are behavioral differences. But you can see, this lineage continue to diverge, is what I was going to say. And so we shouldn't expect to see bonobos evolve into chimps at any point. Instead, these are going to continue to branch off, right? And some of these branches might not be successful, might not make it very far, and other branches might continue to move into other territories and be successful. So just an example of the types of diagrams that we will be drawing, how we will depict evolutionary relationships, and how it helps address some misconceptions about evolutionary relationships like the ones that I had in the past. What else? There is really only one other thing, and it's really just kind of a fun thing, and that is just to look at some pictures. This is similar to what I did last semester. I brought in a bunch of books, and that was because this is a science majors class, and I wanted to introduce you to some influential books that you might find helpful as you navigate the courses that you take as a biology major. And this is one of the books that I would have brought last semester had I had it. But this used to be one of my favorite books. It's probably 20 years old now, that I used to carry around. It's not a religious book despite the title at all. It's just it's a great coffee table book. I'll just take that thing off. How about that? It's a great coffee table book because it's just a book of pictures, right? And what's interesting about it is that each time you flip a page, as you go from the front of the book towards the back of the book, it zooms out in scale, right? So this is all about studying the scales of nature. And what's really interesting, like right off the bat, what you start to see is, we'll see a couple of those things. This from a distance, that's silicon crystal structures, right? Look at that from a distance, and then look at this next page, which is a cross-section through skeletal muscle. You start to see some similarities at drastically different scales. And that's what's interesting. That's one of the things that I think is fascinating about studying any particular scale of nature. When you zoom out or you zoom in, you start to see these kind of repeating things, these similarities at different scales of nature. And that's why studying one scale of nature can give you insights to how the other scales of nature work. And with just looking at pictures real close, it's sometimes not obvious what we're looking at. All that we're looking at here is a few of these pictures that I took from this book that I've put up here on the overhead just to kind of show you. And really, it goes all the way out to like deep space. And what I liked about this book is that it got me thinking about different scales of nature. This is one of the first pages, the first pictures in this book. It's a famous picture from, it's one of the first images that they got inside those particle accelerators. They were called bubble chambers, where originally, I don't have a periodic table of elements in here, but last semester we got into the atomic nucleus, and we said there were these tiny little electrons that orbit around the nucleus. But inside the nucleus of an atom, there's the protons and the neutrons, and we didn't get any further than that. But there was a lot of questions about, could you smash electrons and get anything smaller than electrons? You couldn't really find any way to smash electrons and get smaller than that. But the protons and the neutrons, you could smash and get smaller particles that come out of that. So this has led to the discovery of quarks, and there's been several different particles that have been found since then. Not that this is within the scope of this class, but it's just a very small picture. As we zoom out, we start to see things like this. Like what comes to your mind when you see this picture? Does that? I thought of rivers, I thought of wetlands, and what's happening in wetlands, you've got a bunch of these creeks or river tributaries, and in wetlands, the soil there kind of filters out a lot of the pollutants, so that the water is cleansed before it reaches the ocean. This is a liver cell inside your body, and the liver cells do the same things. They process things that you put into your body, and they filter out some of the toxins. So a similar function, and we see a very similar structure at drastically different scales. There's something about that that's fascinating. This to me looks like somebody's hair that they need to comb out a little knot of, but this is actually just a collection of collagen fibers, the same tissue that you would find that makes up your ligaments and your tendons that hold your muscles to bones or hold bones together, but it looks like hair. This is, we're looking at somebody's lung that has had the tissue dissolved, so that what we can see is just the bronchial tubes that deliver air down deep into the lungs. You can see kind of that branching nature of the lung. Doesn't look too different from the branching nature. This is a satellite image of the Mississippi River Delta. You can kind of see that fractal geometry of nature. This branching creeks and river tributaries looks a lot like the branching structure of the bronchioles in the lungs. This is an organ inside the body. This is a satellite image of a portion of the earth. Here we are back in the body again. This is a cross section through the kidneys, and the kidneys are going to filter the blood for to get toxins out, or maybe not just toxins, all kind of things that the kidneys are removing from the bloodstream. But all of these tubes here are going to process that filtrate, lead it into this ureter and take it over to the urinary bladder. But structurally, this branching nature looks very similar to other things that we see in nature. Are we inside the body here? No. This is once again a satellite image of the Colorado River Delta. I don't know why I couldn't get that out. This is the Gulf of Mexico. They've obviously done some color changing to this to take the green color away from the land and make it look a little bit more brown. But again, you can see this branching pattern, this fractal geometry in nature. It looks just like the branching of blood vessels in the body, or even the branches of a tree that are growing. We'll see this as well when we get out and we start to classify. It's very interesting that each individual tree, right, even trees of the same species, they all have this fractal geometry. They have these repeating things. Like, I know that I'm just using words here. This is not a real example, but if you have like an oak tree or a particular tree or something that you can go on walks and see, if you'll notice that sometimes trees will have like a weird pattern where like a branch will ever so often come off at a weird angle. And if you follow that branch all the way up through a tree, you'll see that same little weird pattern repeat itself over and over. This is what we mean by the fractal geometry of nature. And as we get to larger scales, we'll start to see this. We're zooming out even further. I'm just kind of going a quick flip through a lot of these pages. And now we're seeing, it's not hurricane season, but you can see these areas of low pressure that if they get any kind of spin to them, they can start to form these hurricanes. Anyway, as we zoom out, you can see that here now we've zoomed out to where sometimes there are a couple of these areas of low pressure that start to spin and form these tropical storms or hurricanes. And those areas of low pressure the physics are the same, right? It's drawn together, and as these two storm masses merge, you can see that it draws out these long arms of clouds. And that's not too unsimilar to, if we keep zooming out here, in extremely deep space, we start to see these galaxies that have a very similar spiraling structure. It's not an area of low pressure, but instead it's a supermassive black hole that we have at the center of all of these galaxies. And those two, like that area of low pressure, have a pulling force on anything that gets too close to them. And just like those weather patterns that we're smashing together, we can see that galaxies can and do smash together. This is a picture of two that have just merged recently. You can see their two supermassive black holes have just passed one another. All the planets and the orbits have been kind of ripped apart. Galaxies Merged, which is, I'll show you just a couple of more pictures. Here's a picture after, probably millions and millions of years after the initial merge, and these kind of slowly end up condensing to one big super galaxy. There's actually evidence that our galaxy, our Milky Way galaxy, has already in its past merged with other galaxies. So some of the stars in our galaxy are from a completely different galaxy at one point that has now merged together. There was some NOVA documentary that I saw over the break that was recent updates on all of that. Again, this happens on such a long time scale that we are not around to see this. So you might wonder, how do we even know anything about galaxy formation or life cycles? And that's because of images like this. This is was a famous image because it was the deepest field image that had ever been seen. This was an image that the Hubble telescope had taken. I forget exactly what year it took this photo, but what's interesting about it is that each point of light that you see there is its own galaxy that contains trillions of stars. And how we know so much about galaxy evolution is that we can zoom in on this little pocket of galaxies right there, and you can see two that have just slammed together, right? And then we can zoom over here and see two that have looks like they have slammed together at somewhat of a more distant time in the past, so that they're more coalesced. And then we can look over here, right, and see some that are just about to slam together. So just by looking at different pockets of the sky, we can see different stages of collision or coalescence with these different galaxies. Again, not that this is within the scope of this class, but looking at nature at different scales is interesting. And one of the things that we really want to do here is just get your interest going and find out what scale of nature interests you the most. So this is the last slide, kind of bringing it home to local topics. I mentioned that we're going to talk about habitats, right? And what role in the past, basically, like what geologic forces have led to the formation of the habitats that we have here in Texas, because this really kind of predicates what types of plants that you see. We're kind of right here along this strip of blackland clay, and we know that just by taking a 15 or 20 minute trip west to the hill country, you start to run into limestone and creeks that have clear water that you can walk in. And if you're from the east side of this break, where I'm from over there in Cameron, you'll know it's only a 30-minute drive and you're in flat, clay, muddy, creek-bottomed areas that you really wouldn't want to walk in without some protected shoes. And those habitats that are right next to one another have completely different types of plants. It's all predicated on what types of soils are there. That determines what types of obviously plants and what types of animals. So I'm going to spend a little bit of time on Monday kind of setting up how Texas got to be this way, and some of the abiotic conditions that are going to set the stage for the organisms that we see. That kind of ties into our Earth history stuff on Wednesday. But with that out of the way, obviously I'll just want to say thanks for coming to class, and want to make sure that you know how to find stuff on D2L. You know how to get a hold of me if you have any questions. And at least you have an idea of how this class is going to go. Remember, there's no lab today. There's going to be no lab next time. I feel like that should just be in a couple of days, but I have to keep reminding myself that it's Wednesday, so I won't see you again until next week on Monday. So watch for a second PowerPoint to show up on D2L. That's the new one that I'm creating now, that just has a bunch of pictures of different ecosystems and talk about populations and communities. Everything else is already up there. But if you run into any questions, feel free to reach out and send me an email. That's it. I guess that's where I'll close today off.